When you send ticks into the INHS Medical Entomology Lab, they are identified by species and stored in vials of ethanol until they can be tested for pathogens. We start the process by laying out all the ticks in order to allow the ethanol to evaporate. This is important because the ethanol can interfere with the digestion of tick material later in the process. We then transfer the ticks into bead tubes. The first stage of the process is crushing the ticks, which is done on the tissuelizer. This machine shakes the tubes back and forth as fast as 30 times per second, causing the beads inside the tubes to homogenize the samples. As the ticks are crushed, the DNA content of the samples and any pathogens they may hold are released. The specimens are then spun in the centrifuge, pulling everything to the bottom of the tube and extracting additional DNA out of the homogenized material. Next, they are put in the dry bath, where the temperature is increased and held constant to allow further DNA release. After this, the liquid from the tube is transferred to a deep well plate and loaded onto an automated DNA purification instrument called the Kingfisher. Multiple wash buffers are also loaded onto the machine to ensure that no contamination of the DNA occurs. The Kingfisher starts by mixing the liquid homogenate with digestive enzymes that remove any remaining tissue. After this, we add a binding buffer and a solution containing magnetic beads that attract the DNA. DNA is returned to the machine. Here, it is washed by the wash buffers to remove any residual contaminants. The Kingfisher finalizes the process by releasing clean DNA into the elution buffer. The elution buffer containing the DNA is removed from the machine and carefully sealed. Finally, the liquid is sent to the University of Illinois Biotechnology Center for PCR testing. PCR testing is able to detect the DNA of a wide range of pathogens in a short time span, while ignoring the DNA of the ticks.